Shamirullah, you can start. We are live.
Okay. Good morning, all of you, the respected speakers, all students, and all teachers who are participated in this international webinars. Welcome to today's webinar organized by Department of Chemistry, Shampur Siddheshwari Mahavidyalaya, in collaboration with ICAC, Shampur Siddheshwari Mahavidyalaya. Today's webinar topic of discussion is about chemistry, human life, and challenges to the student under the COVID-19 catastrophe. I would like to introduce today's presenter. Today we have with us Dr. Dipankar Mandal, Dr. Subhrangshu S. Mandal, Dr. Soikat Chakraborty, and Dr. Malabendu Jana. Now we are going to our webinar with the inaugural song. The inaugural song is presented by Riya Hajra, a BSc student of our college. Please, inaugural song. Thank you, Riya, for presenting these beautiful songs. Now, I am requesting HOD of our chemistry department, Dr. Sukumar Guchait, sir, to say a few words about this webinar.
Good morning to all the participants and the respected speakers, Dr. Dipankar Mandal, Dr. Subhrangshu S. Mandal, Dr. Soikat Chakraborty, Dr. Malabendu Jana. Dear students, respected principal, honorary members of governing bodies and ICAC members of Shampur Siddheshwari Mahavidyalaya, dear colleagues, non-teaching staffs and students representative of our college, I hereby take the opportunity to welcome everyone on behalf of Shampur Siddheshwari Mahavidyalaya, Ajodhya, Howrah, West Bengal, India, participating in the one-day international webinar on chemistry. based on chemistry, human life, and challenges to the student under the COVID-19 catastrophe, organized by the Department of Chemistry in association with Internal Quality Assurance Cell, Sampur Siddheshwari Mahavidyalaya. The outbreak of COVID-19 has restricted everyone under lockdown and forced every person around the globe to endure many difficulties which have led us to think in a new dimension. The common people, daily wage laborers, health workers, police personnel, students, teachers, as well as many others from different walks of life have faced several unprecedented challenges due to this pandemic. A complex and unexpected outcome of the lockdown had induced many negative impacts in human mind like frustration, boredom, etc., leading to unexpected situations. Many challenges to human civilization have arisen in the rise of the pandemic. The education sector has witnessed major transformation in the last few months. Being a developing country, India is facing major challenges in spreading awareness and education to different remote areas, which lack infrastructure and network facility. However, viewing from a positive side, the situation has unified all, irrespective of social, economic, and ethical differences. The teachers play a vital role in the society and has a responsibility towards others for providing a way to overcome the hurdles. They are trying to convert the challenges to opportunities for future generations. The teachers are engaged in proper blending of online and online modes of teaching using science and technology so that everybody can carry on their enrichment and upliftment of knowledge even in the face of this crisis. Our faculty members are working in converting and adjusting with the curriculum of online delivery and adopting with teaching for online mode. We have embraced the recent changes and are eager to take the risks of online teaching and learn the pedagogical use of technology. We observe that development of digital learning to serve remote communities was initially seen as a gradual and perhaps arduous task, but situation has helped us to fast forward at present, which is matter of necessity. In this context, we have organized this webinar to spread awareness and knowledge to all. We know that evolution to civilization all had been associated with chemistry. The progress of chemistry helped humans to be self-sufficient in food, has provided solution of health-oriented problems, has given us various comforts of life, and has served in almost every field of life. I believe that chemistry has a vital role to combat the situation arose by COVID-19. For about few months, we, the people, have come across so many new terms like pandemic, COVID-19, quarantine, new normal, etc. And slowly we have accustomed ourselves with these terms. Of course, every household has their own story and gradually being lost with so many thoughts to waking up every morning with the hope of new beginning, we have moved on. Today, we are here together presenting the webinar, Chemistry, Human Life and Challenges to the Student under the COVID-19 Catastrophe. Hope you will enjoy it. Now, I am handing over this to my fellow presenter, Professor Samirul Islam.
Thank you, sir, for your valuable word. Now, I am inviting to our respected principal, Dr. Santu Kumar Bose, to say few words. He is unable to come on live now due to his busy schedule. He sent a valuable message through video. Please, Debuda, please show the video.
Good morning, everybody. First of all, I welcome all the participants in the international webinar on chemistry, human life, and challenges to the students under the COVID-19 catastrophe, being organized by the Department of Chemistry in association with Internal Quality Assurance Cell of our college. I would like to convey thanks to the honorable speakers of this webinar, Dr. Dipankar Mondal, Postdoctoral Fellow, New York University School of Medicine, USA, Dr. Subhrangshu S. Mondal, Professor, Department of Chemistry and Biochemistry, the University of Texas, Arlington, USA, Dr. Soikat Chakraborty, Visiting Assistant Professor, Oxford College, Emory University, Georgia, USA, and Dr. Malavendu Jana, Assistant Professor, Rose University Medical Center, Neurological Sciences, Chicago, USA, and they will deliver their valuable lectures in this webinar. I would also like to convey thanks to the coordinator and members of the Internal Quality Assurance Cell and the teachers of the Department of Chemistry of our college who deserve it for arranging such an international webinar in this time of pandemic. I think the participants will be benefited from this webinar. I expect a grand success of this webinar. Thank you all once again.
Thank you, sir, for your valuable speech. Now, on behalf of ICAP member, I requesting to Dr. Srikant Nondi to say a few words on this webinar.
Myself,
Please, Dr. Nondi.
Dr. Srikanto Nondi, IQAC representative of Shampur Siddheshwari Mahavidyalaya. First of all, I would like to extend my thanks to our respected principal sir, Dr. Santu Kumar Bose Mahasaya, all members of the governing body, all members of the Internal Quality Assurance Cell, and all members of the technical support team. Without their untiring involvement and support, this international webinar could not be possible in this critical time of COVID-19 pandemic. I would like to extend my special thanks to the IQAC coordinator of our college, Professor Rajarshi Mukherjee, as he has been successfully coordinated. Now, I welcome all participants present here, as well as our esteemed resourceful speakers of today's webinar. Hope all of you will enjoy today's webinar. Thanks to all. Over to Samirul.
Thank you, Dr. Nondi, for your valuable word. Now, without taking much time, I would like to request Professor Debojyoti Bhanjo to introduce our first speaker, Dr. Dipankar Mondol. Please, Professor Debojyoti Mondol.
Thank you, Samirul. Let's start our discussion. Now I would like to introduce Dr. Dipankar Mondol, who is the first speaker of this webinar. And today, his topic is what happens after alcohol consumption at molecular level. Dr. Dipankar Mondol is now work as postdoctoral fellow at New York University School of Medicine, USA. He received his bachelor degree in chemistry from Jadavpur University in 2008 and master degree in chemistry in 2010 from IIT Kanpur. Dr. Mondol received his PhD degree from IIT Kanpur under the guidance of Professor Debabrata Goswami. He joined as postdoctoral fellow under guidance of Professor Nilmani Sarkar at IIT Kharagpur, and then under Professor Timothy Leonette at New York School of Medicine, USA. Dr. Mondol has published his original research paper in reputed international journal. He is an experimental expertise in the field of optical tweezers, fluorescence, upconversion, two-photon fluorescence microscopy, fluorescence spectroscopy, etc. Dr. Mondol has received various prestigious awards like National Merit Scholarship for 2003 to 2005, Optical Society of America, OSA, USA, for International Student Travel Grant Recipient 2015, Science and Engineering Research Board, ACRB, India, for International Travel Award 2016, Society of Photo-Optical Instrumentation Engineering, SPIE, USA, for International Student Award 2016, Now, I would invite Dr. Mondal to deliver his speech. Dr. Mondal, over to you. Please.
Thank you, sir. Thank you for your kind introduction. It's my pleasure to present here. I work in like optogenetic technology here in New York University, but today I will talk in more general way, and this topic has also had deep connection with my current work. So the topic of my presentation is what happens after alcohol consumption at molecular level. So alcohol consumption is like common in like East Asia and India also, and there are a lot of like bad effects and what's going on inside our body, what really happens at molecular level, we don't know. So that's why I decided when I got like invitation to present my work, then I thought this must be a good topic about chemistry and human life. So I will go through all these like outline. My presentation would be, I'll first tell about how alcohol metabolism effect on gene oxidation, because alcohol initially oxidize, and then there are a lot of reaction step happens, and it finally go to gene expression contributes. So what is gene expression? I do work in gene expression field. So I would say about what is gene expression. I will use one or two slides about that to explain that. And then I will explain about how gene expression control living body. And then I will tell about how alcohol metabolism actually have like bad effect on our body. And that is like, it's an immediate effect. And the gene expression, which is like long-term effect. And then like how this metabolism gene expression, through which pathway it happens, and finally it actually modulates our brain function. So I will go through one by one, and I will go like every step and try to explain. So first of all, there are some horrifying news, and that astound West Bengal. It happens around 2011, and it was like very nearby place from my hometown, and it was like very toxic. Alcohol kills a lot of people, and it was like kind of a few months it was in headline in the newspaper. And then that time I don't know all much about that, how that happens in human body and how people are dying about that excessive alcohol consumption. So later when I came here, I was doing some epigenetic or genetic technique development, and then I'll get to know what is the bad effect and what is the long-term effect of alcohol. So last year I got a Nature paper, and I think it's a landmark paper in the field of gene regulation, which connects the metabolism. So this paper entitled Alcohol Metabolism Contributes to Brain Histone Acetylation. And what is histone, and what does mean histone acetylation? So I will go through all this step next few slides, and you will get to know how finally alcohol consumption effect on our body. And I also feel about how the alcohol metabolism like have like bad effect on our liver cancer, or like other bad effects happens in our body. And also it has gene expression, which in sense this paper reveals that it's not actually bad. So this is the pathway alcohol metabolism cycle from oxidation to gene expression. First of all, in the first step, alcohol is oxidized to aldehyde by alcohol dehydrogenase. In human body, there are various kind of alcohol dehydrogenase is present. And if you consume, or if we consume alcohol, there will be a lot of aldehyde production in human body or any eukaryotic cell. And then aldehyde to acetate formation happens by aldehyde dehydrogenase. And this enzyme is basically, it's really important if people are consuming alcohol, if inactivation of aldehyde dehydrogenase cause cancer. So effectively, the high concentration or highly gene expression of aldehyde dehydrogenase in human body can inhibit the cancer. So the third step, which is the acetate to acetyl-CoA formation, which happens via acetyl-CoA synthesis 2, which is another different enzyme. And this enzyme is present in liver as well as human brain. And finally, the next step, it's not final. Just before the final step, the acetyl-CoA, the acetyl group, CH3CO, it's a very small, like those are doing chemistry degree, they know it's not a big molecule. So there are a lot of complex reaction, but it's a simple reaction. It's CH3CO group. It will be only added to a lysine protein by other kind of histone acetyl transferase. There are a lot of different histone acetyl transferase. I'm not going to spend a lot of time about that. So I have given only a few examples of P300 or GCN or CBP. And then this histone acetylation actually directly connected to gene expression. So I typically work in the last two steps, and I will go through all those, but I will go through all those steps, how it happens and how human body or like eukaryotic cell, this step by step it happens, and finally it leads to gene expression. So this is how is gene expression. So typically the gene expression does mean there is some information inherited in the DNA sequence, and that inheritance sequence is finally transcribed to a certain kind of protein which helps to function like a human body. Like I can give like few examples, like in our eye there is kind of receptor protein, so it like accepts light, and then we can see. There are like pepsin, it like during digestion it helps, and there is like kinesin protein, it helps like to move our body. So there are a lot of different proteins in human body, and each and every protein has their own purpose, and they are produced a certain amount in every body, and that actually makes like every body different. So the gene expression pathway, one of the like fundamental like step is from DNA to mRNA production by RNA polymerase. This is also one of the enzymes. So from the right hand side, you can see a G file where this is the DNA sequences and going through the RNA polymerase, and RNA polymerase read that sequence and it forms the complementary mRNA. And then mRNA, there are like each and every chemistry student may know, like there are three DNA, they each have that one amino acid sequence, and then amino acid form protein. So this is the basics of gene expression, from DNA sequence to protein formation, and which that protein like help to functionalize our body for different like purposes. So how does gene expression look like? So I do work in single molecule imaging, and we try to modulate the gene expression. Like if we want to express certain kind of gene, if we try to produce their amount, and if we have like bad genes, like there is some oncoprotein which actually generates cancer in human body. So I'm not going to detail how they produce cancer, but we can like modulate and decrease their gene expression. So this is a cell line we like get from this group, the paper I have mentioned, and we are like experimenting. I just give one video to show how gene expression look like. And it's not like in our body, the protein or like DNA sequence to protein production is not continuous, like all the time it's not producing protein. It has certain rhythmic nature, like when it requires how many protein needs to be prepared in per second or per minute, when it needs to be increased expression, when it needs to be decreased expression. It has some certain rhythm, and nature has done it like excellent way. So I would say, so here there is like different cell lines, and one of the protein is marked with fluorescence molecule. Those of don't know about fluorescence, I can go one slide and then I can again come back this slide. So fluorescence, what is fluorescence? So this is a like basics of chemistry. Like there are molecules, if you give energy, they will go to excited state, which is called absorption. So molecule, if you absorb energy, it is not like that they will stay all the time in the excited state. Then it will like spontaneously, it will relax back to the ground state. But it can happen in many ways. I am going only one like way, which is spontaneous radiative emission, which is fluorescence. So molecule can, and they can emit the energy, but obviously that energy is lesser than the energy it absorbs. So because there will be like non-radiative relaxation, there is internal conversion, intersystem tossing, so that energy lost some. So the energy we given to the molecule and we are accepting, that is lesser. So this is the Jablonski diagram. It is the common textbook diagram you will find in the third year BSc. So I can again go back what's the fluorescent helps to elucidate the gene expression. So here is that molecules, which is a single protein molecules or single locus, and it was tagged with some protophore molecule. So that technology is a bit difficult, bit complicated. I'm not going to explain that. So I can only want to show.
Excuse me, sir.
Yes.
Do you want to share your screen?
Yeah, my screen I have already shared. Is it not shared?
No, no.
Oh my God. Stop. So I would go back to first slide or
It's okay, sir.
okay, now it's working.
Okay, okay.
So I'll go from first slide or like where I was.
Okay, sir, you can continue your lecture.
Okay, so I was there. These are the steps I already told that that happens during alcohol metabolism cycle from oxidation to gene expression. I have already explained about what gene expression is and how the gene expression look like in the right-hand zip file. You can see. So from DNA to mRNA, then amino acid, and then protein formation, which is like gene expression. And the gene expression, how it does look like, I was going to show you one video. So here is each dot here in the every circle is represent a single cell nucleus, and each dot you will see white color, which is a single locus or single gene or single protein is producing. So here it's not seeing protein, we are seeing mRNA, which is the intermediate step of the of protein. So here I can play and you see the gene expression, how the intensity varies here. So the cell is moving, that's why it's not exactly same at the nucleus was plotted. So you can see here the intensity profile of the gene expression. It's not like that it is continuous. Sometimes it's on and sometimes it's off. So this actually rhythmic function, it's like it's control the human body formation. So how does that happen? So we know in the basis of our human body is some DNA, and DNA form is some adenine, guanine, cytosine, and thymine bases. So they form the chromosome to DNA, and that sequence is read, and then they form the different protein that inherited in the nucleus, and then cells form the tissue, and then tissue form the organ system, and then total organ system form a organ system, like one organ to organ system, and then different organ system built a human body. So this is our human body is from molecule to different step. So now how each human body is distinct from one to another, how does that happen? So that happens due to the gene expression, amount of gene expression each cell. So if I show you one example here, it's a stem cell. If one kind of gene is expressed high, when it differentiate, it is divide, then it's a gene A, it is highly expressed, maybe gene B is different. I would say there are a lot of genes in the are expressing in the nucleus. So if a high expression of cell give a cell state which is different compared to if cell is expressed gene it's a mistake, gene B. So then the fate of the cell would be different. So that's how the body is developed during our zygote formation after that. So there is stem cell after differentiation and different expression level, they form different body parts. and that's how our body is built. So gene expression has very important role in human body formation. So now I'm going back to the metabolism step from alcohol, and then step by step we go how that is finally going to gene expression. The first step is like alcohol to aldehyde formation, so which mediated by the alcohol dehydrogenase. So alcohol dehydrogenase is an enzyme which present in eukaryotic cell, and it's a like gene dependent enzyme. It takes one molecule of NAD plus and it forms NADH. And finally, I am not going to like each and every bit of step. There is mechanism is given, and I have taken the PDB file from here. PDB code is given here. Sorry. So this is like each and every step. One molecule of alcohol form one molecule of aldehyde. And now the like crucial question is why, like if we see in India or maybe like all over world, in most of the cases you will find the alcohol mediated cancer happens in liver. Why does that happen? Because if we go through the gene expression level of this enzyme particular, which oxidize alcohol to aldehyde, which if we see in different cell type how they express, we will see the expression level of this gene is very high in liver. So you can, this is the like expression level of different cell type of this gene. So this is ADH4. It does mean there are several types of aldehyde dehydrogenase. It is one type of aldehyde dehydrogenase, and its expression level is very high in liver. So it is obvious the alcohol will metabolize in liver, not other cells, because the enzyme is present here. There. So I have also present. There are few other ADH. There are like various ADH1B, which is present in other different cells. But most of the ADH, there is also another ADH gene expression, which is like mainly expressed in liver. So it is very obvious most of the aldehyde will form in the human body in liver, because this metabolism or oxidation pathway is happening only in the liver. And that data was taken from this portal. And now the second step, which is like aldehyde to acetate formation. This is like this enzyme, which is very good for our health, because it was observed that like most of the like liver disease or liver cancer happens inactivation of this enzyme. So this is ALDH, aldehyde dehydrogenase. So there are mainly two kind of aldehyde dehydrogenase, very active aldehyde, ALDH2 and ALDH1, but there are 19 ALDH in our body. And this is the mechanism by which aldehyde to acetate formation happens. There are some active site like glutamine 399, lysine 122, and these are like mechanism. I'm not, again, like you can see this each and every step here is also like similarly NAD plus is added, and then aldehyde is formed to acetate. And these are the this is only the picture was given active side of the aldehyde dehydrogenase. And now we will see how this aldehyde dehydrogenase is present in our different cell type. So because if our body in liver, there are alcohol is metabolized from alcohol to aldehyde. So aldehyde is not good for our health because it's a very reactive species. It can go both way. So it needs to be converted in other molecule which is less reactive. So this is the molecule which is aldehyde dehydrogenase, which form aldehyde to aldehyde acetate. So we need to see in different cell type how this enzyme is present. So if we see, there is actually substantial amount of ALDH2 is present in liver. So it is obvious that it would not be bad for us if someone consume alcohols and it metabolites aldehyde to acetate. But it was actually observed that inactivation of this enzyme, because every human body is different. There is probably this gene is inherited in DNA and that code is inherited, but how much that protein would be produced in our body or in liver cell, that is very different or very diverse from human body to human body. So it needs to be, everyone should be careful how their body is working after alcohol consuming. It has some like stress or like reactive oxygen species generation in human body. That because there is less amount of ALDH is producing. So there is also like ALDH-A1, which is other type, which is also have like higher expression. You see liver, this is like liver, there is a high expression, and also other kind of LDH, there is also high expression. Other body parts also have LDH1, but typically alcohol as metabolites in liver, because the alcohol to aldehyde production happens in liver. So we mainly like how it is active in liver, that depends. So when alcohol metabolite can cause cancer. So today there is like 19 LDH that I have already stated, and there is like two most important, LDH1 and LDH2. And this LDH not only involved in like in this alcohol metabolism, there is also other cellular function, and like inactivation of aldehyde, it causes cancer. And it is like reported that 80% of the world population and approximately 30 to 40% of population of East Asia, and it's like very high number, which carry inactive LDH2 gene. So inactivation of gene, you know what will happen after alcohol consume. So be careful about that. Now this is the third step, which now the acetate to acetyl-CoA formation, which takes place by this enzyme, which is acetate-CoA synthetase 2. And this particular enzyme is not only present in liver, but also present in like brain. And they are in an active state. It's not like earlier case we have stated LDH is inactive sometimes, but it are active in like brain and as well as in our like liver. And this is the like different for like pathway how the acetyl-CoA forms. So there is also not only alcohol produced acetyl-CoA. So if we intake glucose, there is like other pathway they can produce acetyl-CoA that finally take part in the gene expression. So I'm not going to that to explain how glucose produce that in the mitochondria acetyl-CoA. I will only focus on this talk how the acetate which was produced from alcohol that finally contributes to produce acetyl-CoA, and that finally helps to acetylate histone, because their acetyl-CoA also can form the fatty acid, and fatty acid also have diverse role in membrane formation. It also have like a strong role in the metabolism, different metabolism pathway. I will only explain how it contributes to the acetylation of histone protein, and that finally leads to gene expression. So these are the expression level of ACSS2, what I was already stated. So the expression level of ACSS2, which is liver, is like higher, and which is in brain cell, there is not that high amount, but there is like little bit of present, and it is sufficient to like metabolites acetate to acetyl-CoA. And then final, like before final step, this acetyl-CoA, this acetyl group is transferred to lysine, and lysine of histone protein. So next few slide, I probably explain what histone protein is, what does histone acetylation mean, and how it finally contributes to gene expression. So this is also happens due to one enzyme. So in like chemistry courses, everyone know what enzyme does mean, how enzyme kinetics is. It is like chemical kinetics chapter. If you go through, you'll get idea enzyme, how does work, how the enzyme mechanism is. I do work on these final two steps, and I will like explain how this formation of acetyl-CoA from alcohol, or maybe from glucose and from other like metabolism pathway we are getting in our human body, finally leads to gene expression. So this is like, I will now explain about, like I talked a lot more about histone. So what is histone protein? So if you like think about eukaryotic cell, so there is like typically 6 billion base pair. So how long that will be? So each base pair length is typically about 3.4 angstrom. So if you then like multiply it by 6 billion, it would be kind of 2 meter length. So in our body, each cell has like total DNA that is length of 2 meter. And now consider each cell dimension is maybe like height 10 micron, width 10 micron, and like height is 10 micron, which is typically 1000 micron. So can you believe it is possible that DNA would accommodate in that small amount? It's never possible. It's never possible that 2 meter length of DNA to accommodate on that nucleus of 1000 micron volume, micron cube volume. So what happens and how it stays in that nucleus? So it's a kind of good kind of like mechanism, but how it developed, I have not like that good idea about, but I know the mechanism how it does. So what happens, I think like everyone knows in like India, in like West Bengal, the kite flying, there is kind of in Bengali called lata. Like if you wrap your like wire in that things, and you can wrap, and there is like histone protein kind of that. The DNA are wrapped around the histone protein, and there are 4 or 5 kind of histone protein: H2A, H2B, H3, H4, and H1. It is 5. There are actually 4 important here: H2A, histone, like histone 2A, histone 2B, histone 3, and histone 4. They form an octamer, and then DNA are wrapped around those, and they wrapped 163 base pair of each histone, 147, and there is some linker. So 163 base pair. You can like after 1 octamer there is 163. So it is like there are a lot of histone proteins, and DNA are packaged around these histone proteins, and they can now like happily accommodate in the nucleus. Like you can believe now that 2 meter of DNA can accommodate in 1000 micron volume of nucleus by wrapping around that protein. So it helps. But there is also thing, if your histone protein is wrapped the DNA, how you think the transcription or maybe gene expression would happen? It never possible, because if something is not accessible, then how can it will be usable? If you're not accessible, it is not usable. Like if like DNA are packaging around histone protein, then RNA polymerase cannot go enter, and then protein would not be formed. So there needs some space. So it's not only good for some like all the time. So histone is wrapped around, like DNA is wrapped around histone, but sometimes it's need to be unwrapped, because it needs to be read by protein, so that RNA pol2 protein, so that other kind of protein can be expressed. So here is the 2 other enzyme is present, which back and forth this like mechanism, like one protein helps to wrap around, one protein to release. So what is the mechanism of that? So DNA is kind of phosphate backbone has, and then lysine protein, if acetyl CoA is added, then this acetyl CoA is negatively charged, which is like repulsion. It's unwrapped, and then it is accessible for the transcription and gene expression. So here is like a picture was given unwrapped. It is cryo-electron microscope image, and these 2 like enzyme, histone acetyl transferase and histone deacetylase. That's why it's important for gene expression. So each and every bit of enzyme I so far explained, important for do their work in every bit of step. So one of the enzyme inhibition may impair the gene expression, and it may cause like deadly disease, sometimes maybe cancer. So this is the mechanism by which histone acetyl transferase added like CH3CO group to the protein of histone. So histone is one protein, so there are a lot of backbones. So there are like methyl group can be added, acetyl group can be added, and then phosphate group also can be added. So these are called post-translational modification. So those I am not going to explain. I'm only telling that methyl group can be added, that can give other kind of gene expression, like acetylation can be happen, and that can also give like different mechanism of gene expression. So this is the like mechanism how acetyl group is added to the lysine group. And now I would say about if the acetylation doesn't happen, how can it affect? So here you can see, and like in every, like I think like everyone know, this is the cucumber tree, and this is called tendril. So everyone see, probably we can also see in our home, like in pumpkin also, this tendril. This is called tendril. So this is wild type. It is common, and it's wrapped around some support, and the tree grows. And if some way the impairment of this hat of happens, how would be the fate of this tendril? So there is like very complicated mechanism, the CRISPR-Cas9 technique, which was now very like common in USA, people are using, and India has just started to using that technique. that way they have impaired the HAT. So what is HAT? I told already. This is the histone acetyl transferase. It is an enzyme which transfers like CH3CO group to a protein. So if we impair that, then you can see this, like this cucumber tree compared to this cucumber tree, there is no tendril, and there is, if there is tendril formation, there is maybe 1 or 2 turn. So there is a it's a inhibition of the HAT activation, making impairment of the cucumber tree. And also thing is that can be happening in 2 ways. So what enzyme which sit on the DNA and then it acetylate, you can impair that, and also you can like modify that DNA which does not love to take that histone, like this HAT molecule, that also can impair. So this is one kind of, if you impair the enzyme, then this kind of impairment happens. And now if you like modify the DNA sequence where this HAT binds, then what happens? Then you will see this is the like normal, there was no modification was done, which is common. You will see in our hometown, like which is like this is cucumber tree in perfect turn. It can like wrap around, it's kind of a wire, and it can grow finely. But if you now modify that binding domain of the HAT, so as the binding domain, if you like modify, then binding would not happen, acetylation would not happen. If then like we'll see there is tendril formation, but there is no turn. So if there is no turn, it cannot support actually. So that is kind of some example. If acetylation doesn't happen, what impairment can be seen. I also do work in this field. I have like tried to enhance the acetylation. So then what happens? So if you see left hand side, it's a control experiment. So I am not giving anything. So it's a normal gene expression. So each dot, like each circular state is in cell, and each dot represents a single gene or single protein. Now it's common, like that's how our body works, how protein is expressed, they are producing, like we can see with receptor protein, we can work by kinesin protein. So that's why. Now I have used some inhibitor molecule, so which actually gives some feedback through different, like there are 2 or 3 like reaction pathway, and then acetylation happen. And we see that there are a lot of cells are now active with the expression level of that gene. So that means if we increase the acetylation, so protein will be produced more. So what protein you want to produce more, that obviously would be good for health, and you kind of oncoprotein you want to like inactivate them. So that's in your hand. Like it's people are trying to now develop different technology to do that, so that like cancer can be can be minimized, because some of the protein also expression level is higher. They can also inhibit that oncoprotein also. So this is, and also one more thing. So if we add that some drug and we see that acetylation is happening, gene expression is higher. So how do we know that is actually acetylation is happening? So you need to know that the acetyl group, CH3CO group, is actually inherited in that chromatin or like nucleus. It was added. So I have some like immunofluorescence. It's a common technique nowadays people use in wide field microscope. In immunofluorescence, I would go first, how is the technique, then maybe I will explain this image and then you'll understand. So this is the histone. So what is immunofluorescence? So let's see, we are targeting here like acetylation. So this is the acetylation we are targeting here, K9, histone 3, K9. So this is, then that's why we need some antibody. And what does that antibody mean? Antibody is some epitope which have, antibody has some epitope binding domain which can bind the epitope or antigen or maybe some proteins. So there is one example. So this is the typical structure of antibody. So which has some like epitope binding domain, which can bind epitope and then can inhibit its activity. So we have used some antibody, so which can bind to a lysine, K-lysine acetylate state. This is like first antibody, which is rabbit polyclonal antibody. And now we want to see, so we use another antibody which have like binding domain of that antibody, and that is bind with some fluorophore. So which is tagged with some fluorophore, and then we can measure how the actually the acetylation happens. So this is the like mechanism. I did the experiment, and I go how the acetylation happens. So this is DMSO control. There is no drug. You can see that very low level expression of acetyl group was incorporated. Like CH3CO group is very less in the nucleus, but if I add this drug, so there will be more acetyl group was incorporated in the lysine. So there is more signal, you see, compared to this. So this is DAPI signal. This is the only DNA signal. So this DAPI molecule bind to the DNA. It doesn't matter on how acetylation is happening. It's like kind of baseline. So you can see, does baseline means that each kind of imaging condition, each cell has similar amount of DNA. So then you can compare one by one. But second image, you can see there is like very few molecules are present in acetyl group. CH3CO was incorporated, but for this, many more molecules. So I already explained this one. So yeah, so I also then like observe, I actually use two different kind of cell line we are using now. So if we activate acetylation, how the gene expression will increase, we see some like kind of increase in the acetylation level. But this all experiment was done like the drug was added when cell are live, but finally measurement was done when cell were dead. But it's kind of experiment you can do, but it's not for like that good sense you can get how the live things is operating on those things. So we are now currently started live cell experiment. So it's very difficult for live cell experiment to do immunofluorescence because you know, every cell has cellular membrane, and this antibody has very gigantic molecule. So 150 kilodalton, it never can penetrate through the cell membrane. Recently, from Japan, Tokyo Tech, like university, they have developed a new technology which is called like Fab production, which is the fragment of antibody. So you can lysis using some protein. You can split in different parts, but still that binding capability of the antibody is still present. So that means if you lysis the protein, there will be like 2 or 3 fragments, but still the binding potential of that fragment would be same as the similar like single antibody. But if you use this antibody in live cell, it would not be able to penetrate through the cell because due to its very big molecule, it is not like permeable through the cell membrane. But small molecule, it's 50 kilodalton, it can easily permeate. So I am now preparing this small molecule based on that technique. You can see if you have like single antibody. So this antibody was in like big 4, there is band like 150 kilodalton. You can see this is Western blot. So Western blot is a technique you can measure the like protein molecular weight and compare with like your like control protein. So I know the protein what I am using, and I want to now check I actually the produce 50 kilodalton or not. So you can see this is the like single protein which we have purchased from the market first. So this is the antibody 150 kilodalton, around 148 or 150 kilodalton. So it was not fragmented. Now I like goes through some of the step, and I have observed there is a band in 50 kilodalton. You see this is Fab, which is Fab, and now we inject those Fab to cell, and now you can happily imaging that one, and we can see the intensity difference if it binds with the acetylation mark. So live cell imaging only can be done with Fab. No antibody can be used because it cannot penetrate through. So this is what I'm like currently doing, and we are measuring the acetylation. And one of the acetylation measurement was given here. If we like activate acetylation, the increase in acetylation rate with time, it's around 3 or 4 hour acetylation goes up. So you can like imagine in our metabolism rate, if we are taking anything, it doesn't that it's like immediately like going to form some molecules. It takes some time. For our case, we see that like its metabolism rate around 3 to 4. It takes 3 to 4 hours. And now I'll talk about like if this acetylation happens through alcohol, it is actually good or it is actually bad. So now I would explain the first paper I have showed, which is like I would say like last few years, one of the landmark paper it published in 2019 in Nature later, which talks about alcohol metabolism, how like modulates gene expression. So there are like mice, they are given alcohol and then was performed different experiment to see it's actually good or it's actually bad. And actually, this experiment proves that it actually helps mice for memory formation. So how does that experiment was done? Let's see. So this experiment was taken from this Nature journal. So first, what happened? We need to know that if mice is consuming alcohol, so how do we know that this alcohol it actually going to liver and then liver to brain? We need to have some marker with that alcohol. So if we eat like normal alcohol or ethanol, it is very difficult. It's a kind of carbon, hydrogen, oxygen, and all those like atom are present already in our body. So it's very difficult to like find actually this carbon, hydrogen is from outside we are consuming that one, or it's already inherited in our body. So what happened? They have given that mice the deuterated ethanol. So ethanol D6 they have given, and then before consuming alcohol and after consuming alcohol, they measure the mass spectroscopy of the hippocampus. So what is hippocampus? I have given that they actually the hippocampus was taken from mice. So hippocampus is one of the part of the human brain, or like any like brain in like mouse brain also hippocampus present. So there is different part, cortex, frontal. So there are different parts of the brain. So hippocampus is one of the part. So this hippocampus was taken and then measured how acetylation was happened. So if you see that deuterium is increased, that does mean that ethanol was consumed, that actually coming from liver and then metabolites, and it caused the blood-brain barrier, then it goes to the brain. So this is like hippocampus mass spectroscopy, left-hand side, before consuming alcohol, that is 0 hours. So there are like 4 peaks. It is like M+1, M+2, and M+3. This is like mass spectroscopy. You can go through and you can go the technology, how it works, like charge based upon the charge, M+1, M+2, they determine the molecule. So here the molecule they are targeting, which is the histone, like histone 4 triacetyl peptide. So and this is like hippocampus mass spectroscopy after 4 hours after consuming that alcohol. So I have also performed experiment. You can see there is 4 hours. This time is also critical. Need to be careful about. So I have also performed this experiment. You see like around 3 and 3.5 hours. So the acetylation goes up and it's kind of saturated. So it takes 4 hours. These experiments also shows that 4 hours the acetylation actually happens. So what happens? So M+1, if you compare like this band like here, it actually goes up here. And this band is M+2, which is here. After 4 hours, it also goes up here. And this M+3, which was here before consuming alcohol, and after consuming alcohol, this goes up here. So that does mean the acetylation does happen after consuming alcohol, and that acetylation group, which was coming from alcohol. So I have already shown how the step by step it was coming from alcohol to aldehyde, aldehyde to acetate, acetate to acetyl-CoA, and then acetyl-CoA to acetate to the human brain, or maybe like when we consume, and it's case for the mouse brain. And now I again want to show you that like this acetyl-CoA synthetase. So here is like a huge amount of contribution of liver and also the brain hippocampus they present. So when someone consume alcohols, after alcohol produce the aldehyde, it cross like in blood, the aldehyde is there. It can go anywhere, so it can be acetyl-CoA formation can form in liver and as well as brain. So that does mean that acetyl group produced in the liver, and it can cross the blood-brain barrier and then goes to human brain. Otherwise, that acetate to acetyl-CoA formation itself can happen in brain because there is present. And they also, like in this paper, they also prove that one. So how this acetylation modulates the brain function. So here is that experiment. So there are 2 kind of room. It was like kind of cage, you consider. The mice was kept. One cage, like one home for mice. So it was given the alcohol to consume. So there is a lot of alcohol, and that is consuming every time. and one room mouse is there, so it is consuming only saline, that is water. So and then it was the mouse was taken and was tested for 20 minutes. So the mice want to stay in that saline room or want to stay in alcohol room. So it was actually like kind of like surprising thing. It was saying that those mice was consuming alcohol, they actually want to stay in that room, and if they consume saline and they have like more diverse role, they want to stay in that room also, they can go to that room. So that means the memory formed when alcohol consumes in that room, some amount of memory was formed because it can remember that I was in that room. So most of the time it's spent in that room. So this data actually that present. So before training, it has no like choice. It can go in the left hand side or right hand side, like white room or like dark room. And when alcohol was given and it was trained and it was observed that it has some preference, it want to stay in that room. So that some memory was developed. And now it's not only that alcohol is directly related to the memory function, because the alcohol is actually going through different steps, and it acetyl group actually, and this acetyl group is formed by this enzyme. So acetyl CoA synthase, like many times I am telling that. Now if some chemical modification, it can knock down. So knock down, what does mean? So in your body, maybe let's say in every hour 100 acetyl synthetase protein is producing. Now what you do, in some way you will make some technique, in that particular time it will produce maybe only 10 or 15, like you will decrease the amount of protein, so its functional activity also decrease. And it has also been observed that if you functionally deactivate that one, and then the mouse again lost that memory, that means mouse has no choice to stay in particular room. So that is one thing, that if you consume alcohols, it needs to be go through proper cycle, and then acetyl CoA synthase must be active, and then it directly connect to your gene expression and maybe form brain function or other also. So there is also a very complicated RNA-seq experiment. I don't want to show that RNA-seq experiment shows that there are like 2000 of more gene was actually some has decreased, some has increased expression, or like they are different kind of protein actually, if I would say. So this is the case, like if your body don't have like acetyl CoA synthase, so this step would not go, so alcohol would not be no use. So that is also like aldehyde dehydrogenase, if it is inactive, if you consume alcohol, there will be liver cancer. It is also that if your acetyl CoA synthase is also not active, your gene expression would not be like affected with alcohol. So this is like maybe final slide. So that's how happened. If people consume alcohol, the alcohol to aldehyde form, then acetate, and acetate to acetyl CoA, and then acetate CoA go through blood-brain barrier, and then it remodel the chromatin, then gene expression happens, and then we behave like different way. So the gene expression is like kind of not only control our body, it also our behavioral change, because like human body, like if we cry, if we laugh, so all are controlled by different protein, the expression level. So if it increases with its baseline, it will like change in mentally also people. So that's the end slide, how that alcohol to brain function, it does work. And now I can conclude with that uncontrollable alcohol consumption causes liver cancer. But it doesn't mean that controllable alcohol consumption is good. If your aldehyde dehydrogenase is inactive, also alcohol consumption is bad, because your aldehyde would not be formed to acetate. So be careful about that. And then alcohol can modulate gene expression, because there is alcohol CoA synthase. If there is no alcohol CoA synthase, it doesn't activate. But one of the also like thing we need to be think about, because I'm also doing gene expression, we are trying gene expression in single gene, because if you want particularly specific about some gene, you want to express some protein you don't want, like I already told that like those protein which form cancer in our body, you want to maybe decrease their expression. Those protein, like some people, they have some receptor protein loss, they are losing their eyesight. You maybe increase that gene expression so you can get back your vision. So that is good. But if you add alcohol, so it will express all the proteins, so there is no control. I don't know what would happen. uh, so there is some RNA-seq data that shows that there are a lot of gene are already enhancing, lot of gene are deactivating. So that needs to be studied. And also this finally alcohol to gene expression happen through histone acetylation. But I have one question still need to be answered, which is that alcohol mediated gene expression can affect our next generation or not. So I presented, I initially when got invitation, I was some planning to a proposal of my faculty application. I was proposing that maybe it also affect our next generation. The people who are consuming alcohol, it has maybe some epigenetic memory. So those are people working epigenetic. Maybe Professor Mondal, will be talking next. He knows better than me. There is some epigenetic memory, and if that epigenetic memory stays during alcohol consumption, it definitely does affect upon next generation. I probably would work with some germ cell which directly connected with generation, next generation formation. And that's finally I will conclude. There are a lot of scope to do with this pathway from metabolism gene expression. It's a very interesting field in this now. People were doing separately. Some people was doing metabolism experiments. Some people was separately, like our lab do gene expression. But now a lot of people are interested how actually metabolism connects with gene expression. So finally, I am concluding by saying that we need to be answered this question: alcohol-mediated gene expression can affect our next generation or not? It's a question. Thank you.
Thank you to Dr. Dipankar Mondal for your precious time and for your presentation. Dr. Mondal has well shared his work to motivate the future generation who wants to know about the progress in current research. He has well presented what happens after alcohol consumption at molecular level and effect on a drinker's health. It is a very important topic, as here is a dark truth behind India's post-lockdown liquor line. After the first lockdown period, it has been seen that there is a long queue where outside liquor shops across the country. Thank you once again, Dr. Dipankar Mondal. Now, I move on to the second speaker, Professor Subhranshu Shekhar Mondal. Professor Mondal is an expert in the field of biochemistry of human gene regulation, epigenetics, and disease. Dr. Subhranshu Mondal is an associate professor at the Department of Chemistry and Biochemistry, University of Texas at Arlington. He received his bachelor's degree from Midnapur College and master's degree from Kolkata University. He received his PhD degree in 1998 from the Indian Institute of Science, Bangalore, under the supervision of Professor S. Bhattacharya. Then he joined as postdoctoral fellow in the laboratory of Professor L. J. Reha Crench at the University of Alberta, Canada. Then he moved to the laboratory of Professor Danny Rinberg to research on eukaryotic transcription and gene regulation in human at the University of Medicine and Dentistry of New Jersey. In 2005, he became an associate professor in UTA. His current research is aimed towards understanding the fundamentals of human gene transcription, and he has published more than 60 original research papers in reputed international journals and has also written multiple invited review articles. Dr. Mondal applied for several patents and presented seminars in various conferences and universities around the world. In parallel to research, Dr. Mondal enjoys teaching biochemistry and interacting with students in the university campus. Professor Mondal, at present, is a member of renowned institute like American Heart Association, ER Focus Group, DFW Metroplex. Institute for Cancer Research and Blood Disorder, American Chemical Society. Dr. Mondal has received several prestigious awards and honors like Ad hoc Reviewer for FLDOH Grant Review Panel, USA, Ad hoc Reviewer for NIH Studies Section, USA. He also received gold medal by Chirantan Rasayan Sanstha, India, Grant Review Panel member by American Heart Association, Mahatma Gandhi Parvasi Samman by NRA Welfare Society of India, Outstanding Research Award 2013 by University of Texas, Arlington. He is also visiting scientist, Indo-Hungarian Research Fellow, Budapest, Hungary. Now I invite Professor Subhanshu Shekhar Mondal. Over to you, Professor Mondal.
I just want to make sure that you can hear me and my slides are on. Can anybody reply in the chat that you can view the slides?
No, sir, your share is not, your screen is not sharing.
Okay.
Okay, now it's sharing.
You sharing now?
Yes, sir.
Can you hear me?
Yes, sir. Yes, sir. Audible.
Yes,
You
sir.
are audible.
Sir, please show your first slide. Okay, okay. Perfectly visible, sir.
Okay. Well, first of all, I would like to thank Dr. Guchait for the kind invitation. And also, I would like to congratulate the college and the university for organizing this fantastic opportunity for us as many students who may be listening to us. And I think at this critical time, it's very, very important for the students to hear what is going on and how to stay motivated and so on. So if you cannot hear anything sometimes, or the slides are not visible, just you can interrupt. I will stop and talk. And if anybody has question, I don't know how the question answer session will go, but anybody can interrupt anytime. So for the today's talk, I would like to say that I want to keep it very simple, considering that there are many college and university level students are there who wants to learn. And so I may not go very deep into the data, what we have. So the talk primarily is geared towards immune response, inflammation, and at the end, I'll touch base a little bit on COVID-19, and we have some of the efforts going in this direction. So the next slide. This actually kind of gives an overview for my lab. I would briefly introduce myself because this is a chemistry department. Originally, I was trained as a chemist and my doctoral degree is in organic chemistry. So I enjoy really the chemistry, but over the years, I started getting fascinated with the mystery of nature, mystery of the biology. And that's how I switched more towards biomedical, how human genome functions and how they are regulated, misregulated in different disease. Actually, my previous speaker, Dr. Dipankar Mondal, already made the stage ready for me. And thank you, Dr. Mondal, for your nice presentation. I enjoyed. So today I will also talk a little bit about the epigenetics and how they are controlled to a variety of human disease and how they are into COVID-19 pandemic and so on, and what are the challenges and how we can help even think of addressing some of the problems. So my lab primarily worked on a series of enzymes which are called mixed lineage leukemia. These are briefly called MLLs, And because based on the name, you can see that they are directly linked to human cancer, which is leukemia. And it happens, these genes which are mutated in leukemia, actually a class of enzyme. They are critical player in histone modification, chromatin modification, gene regulation. So my lab several years from the beginning started looking at the biochemistry of this family of enzyme in gene regulation and cancer, and how we can target them to develop certain therapeutics. Lately, my lab has moved to a relatively new area, which is called non-coding RNA, which I will talk about this today. And I want to make sure that all the students and the listener has some idea, at least the basic concept, what is epigenetics, what are non-coding RNA, what are their significance in human health and disease. In my lab over the last 15 years or so, we explored how these mixed lineage leukemia family of enzymes are involved in steroid hormone signaling. Steroid hormones are critical for variety of health and disease, reproduction, cardiovascular disease, cancer, and so on. So my lab actually has done a significant amount of work exploring the function of these enzymes which are involved in leukemia, in cholesterol metabolism, cholesterol lipid metabolism, to some extent glucose metabolism, more on the steroid hormone signaling area. Because we work on mixed lineage leukemia, we have done significant amount of studies on cancer biology, actually how exactly these MLLs, these enzymes which we work, how they are integrated with the cancer growth, or how we can target them to develop therapy. And lately, because I'm a chemist, my lab is also looking for solutions for some of the human disease. We are screening variety of molecules, either they are synthetic, commercial libraries, or they could be gene therapy to target different type of disease. Also, another area where I am getting highly interested, in fact, at this point, very active in my lab, is looking how the epigenetics, which I will introduce you this term in a very simplified way, how epigenetics is linked to variety of cardiovascular function, inflammatory or immune dysfunctions. So before I get into any of the details, I want to introduce the term epigenetics, and I want to make it in very simple way so that we all understand. We know that genetics, starting from Mendelian genetics 1860s, we know that genetics plays a role how a person look, how a person functions, how a cell. So almost everything is controlled by the gene which we have, the nucleic acid which we have in our body, in our cells. So they are actually blueprint of life. They contain all the genetic information, and this genetic information is copied into the form of RNA, and that RNA is eventually translated into protein, and this protein gives the function and phenotype. So basically, DNA, nucleic acid, has the genetic information which are transcribed into RNA, RNA to protein, and protein gives the function and phenotype. This is the basic concept of genetics, molecular genetics, that all the information for a function, for phenotype of an organism or how it will function, they are all present in DNA, nucleic acids or genome. But when we talk about, we are going to talk epigenetics. When we talk about development of a single organism like human, you start with a single cell zygote, and that single cell eventually duplicates into 2 cell, 4 cell, 8 cells, 16, and eventually they start to differentiate into different types of cell, and eventually fully grown organism like human. So from single cell, eventually all the cells in our body generated. But very important to note that when a cell divides, it duplicates its genome. That means every cell which originated from the single zygote cell, they have all identical genome. That means genetics is identical. So each cell in our body have identical genetics, but it is important to note that even if they have identical genetics, the different cells in our body may have different function, because some cell could be blood cell, somebody could be skin cell, neuronal cell, and so on. They function, their mechanism of action is very different. They are specialized cells. These are called differentiated cells. But they all have identical genetics. So this concept is broadly called epigenetics, that all the cells, even if they have the identical genome sequence, gene sequence, they function differently. This concept is called epigenetics. And this happens because we have certain number of genes in our body. Like human has about 20,000 protein coding genes, and all of these genes are not functional or expressed at the same time. At a given time of the development, some genes are active, means making RNA and protein, some genes are silent, and another time a different genes may be active. So there is although we have 20,000 genes, not all of them are active all the time at a given time. So there is differential array of gene expression and regulation, and that's why different cell expresses different genes. That's why they function differently, they look differently, even if they have identical genetics. So this concept is broadly called epigenetics, and epigenetics means a gene, the nucleic acid genome, is interacting with the environment, whether it is cellular environment or outside the cellular environment. Okay, and that's how the output is done. So environment actually influence or talks with the genome, and environment dictates how genome should function and so on. So this concept is broadly called epigenetics. In my lab, we try to understand the basic mechanism of epigenetics, how gene function, gene expression is controlled by the environment. So with that introduction, we can move to the next slide. DNA, which is well known as double helical DNA, and in eukaryotic cells like human, the DNA is not free, rather complex with variety of nuclear proteins. You heard in the just previous lecture that DNA is complexed inside the chromatins. Basically, DNA are wrapped around the proteins, and there are some histone proteins. I don't want to go in too detail of these stuffs, but I want to give a brief concept. So these proteins have positive charge and DNA has negative charge. So they pack the large piece of DNA by electrostatic interactions inside the tiny nucleus. And this was actually the concept for a long time. But late 90s or so, it is realized that the nuclear positively charged histone proteins, where the DNA is bound, they have much more dynamic function. They get acetylated into the amine group of lysines, methylated phosphorylation in serine hydroxyl group. So there are different types of modification, addition of the small, these groups, acetyl group, methyl group, and these different addition of the groups actually dictates or decides how the gene around that particular part of the chromatin will be functioning, whether it will be activated, that means making RNA and protein, or it will be silenced. And this so basically the chromatin modification actually controls the gene activity. So chromatin modification marks are epigenetic marks, and environment influence, like Dr. Mondal just presented, that alcohol actually controls the histone modification. Surroundings of the DNA, surroundings of the chromatin, that's how the DNA activity is controlled. So environment of the chromatinized DNA is manipulated by these modifications. So histone modification or chromatin modification controls the gene expression, but I think somewhere around 2007 or so, people realized that along with this histone modification, there are a new class of transcripts which are called non-coding RNA. I'll talk a good amount of introduction on non-coding RNA so that we understand what it is at some point of this talk. So these non-coding RNA, these chromatin modification, all together they form kind of epigenetic code. Histone modification, non-coding RNA, the environmental stimuli, all these kind of together works like a code on the chromatin surface. And environmental stimuli like heat, temperature, moisture, nutrients, hormones, they come, enter into the cell, and they influence these epigenetic modifications or epigenetic codes, and that kind of decides overall function of the genome. So that's how, so environmental stimuli, or they interact with the genome to control the genome function. In my lab, we have been studying over the years this function of histone methylation, acetylation, and lately we have a heavy emphasis how these non-coding RNAs are integrated with these chromatin modification mark to control the genome function in healthy cells and in variety of human disease. So one of the histone modification which we are working, or we spent a good amount of time, as I said, is called histone methylation in one of the histone called H3 lysine 4. I don't want to go in detail. This methylation is introduced by these enzymes called MLLs, which I mentioned. These are called mixed lineage leukemia. So these are some proteins which are already known to be mutated in leukemia. About 8 to 10% of the leukemia, especially the childhood leukemia, has something to do with mutation in this gene. And some of these mutations are so severe that very little can be done to this child who has this particular mutation in this gene. So there's a high interest to understand how this enzyme functions, how we can develop therapeutics for treatment of some of these severe cases of leukemia. So that is one class of basically chromatin modifying enzyme, histone methyltransferase control gene activation. We are also interested with another family of enzymes called EGH2 that methylates. That's also an enzyme which methylates another lysine. K stands for lysine, and this methylation is linked to gene silencing, and these particular enzymes are involved in variety of cancers, including breast cancer. In fact, EGH2 is a biomarker for breast cancer. A lot of pharmaceuticals industries and medicinal chemistry lab is spending a lot of time to develop small molecule inhibitors against EGH2 or MLL so that they can be used for treatment of some of these human disease. So we are trying to understand how these enzymes control genome function. In fact, it happens that EGH2, this methyltransferase, also interacts with a non-coding RNA, which we have been working called hot air. I'll talk a little bit on them. So basically, my lab is trying to look how these enzymes control the chromatin modification and how they control gene expression and regulation. So that's kind of overview. I can introduce a little bit on MLL, the mixed lineage leukemia, which are histone methyltransferase. Primary function of MLLs are actually hematopoietic stem cell differentiation. Hematopoietic stem cells are those present in our bone marrow, and bone marrow, inside the middle of the bone has bone marrow, and that bone marrow eventually generates variety of blood cells. Okay, and this happens that this particular protein or enzyme MLL, this is a critical player in differentiation of the bone marrow stem cells into blood cells. That means if this gene or protein has a problem or mutation, the blood cell differentiation will not take properly, and that's a case of leukemia. This is the structure of one of the MLL called MLL1. It has histone methyltransferase. I don't want to go into detail, but this particular gene kind of rearranged, it's recombination events, translocates, and one part of the gene gets fused with fusion. It's a interchromosomal fusion. Like you see the chromosome 11, chromosome 19 fusion, 411 fusion. So basically it's a recombination event where MLL gene is recombined with other part of the other genes in the other chromosome, and this results in malfunction of the normal MLL gene, so which is critical in blood cell production. So once the MLL gene is mutated, people ends up with leukemia. In fact, many times some of the cancer patients, when they go through chemotherapy, their primary tumor is decreased, but they develop secondary leukemia, and often there's a incidence of MLL rearrangement. What causes this MLL breakdown or mutation? It's unclear, but there's a lot of research what drives this mutation and what we can do not to mutate. And most of the time, some of these translocation or mutation is a very, very severe, and that's why there is a high interest. In my lab over the years, we isolated these MLL proteins from the human cells, studied their role in gene expression. We studied their functions, especially in steroid hormone signaling, with emphasis of lipid metabolism. A significant amount of studies has been done on this role of these MLLs in tumorigenesis, hypoxia, and angiogenesis. Briefly, I may talk, and many of these stuffs are published. And in fact, this is a book I edited, I think 2-3 years ago, on gene regulation, epigenetics, and hormone signaling. And this book is very heavily emphasized on epigenetics. What is the basics of epigenetics, gene expression regulation, how they're involved in different types of disease, obesity, or steroid hormone signaling, reproduction, and so on and so on. So I think a good reference for some of the emerging doctoral students to look or learn about epigenetics, gene regulation, and so on. So I will briefly talk a little bit on MLL in cancer. We have done some work. So MLL is primarily known to be involved in leukemia, and that's how it is known. But these days, last about 10-15 years, lot of technologies has come out, and where you can actually profile the gene expression profile, DNA sequencing, gene expression profile, proteomics, all kinds of technology which were not there 20 years ago or 30 years ago. Now it can be done. One can get some tissues from the patient and do their proteomics to study their protein profile. They can do the sequencing to do their complete human genome sequencing to find the mutations and so on. So all of these could be done, and based on them, there are tons of data which is called the cancer genome database. It's a central database, TCGA database, where all these patient data is available. So if you look at those databases, you can actually find that what are the mutations in breast cancer, what are the mutations in pancreatic cancer, whether a gene is overexpressed, mutated, deleted, and so on and so on. So we did some initial analysis on MLLs, a couple of MLLs and other proteins which we are working. MLL1 is primarily mutated, actually rearranged in specific term, but there are many cases MLL interacting proteins, and some of the MLLs are overexpressed in other type of cancer other than leukemia. So we thought, why MLL is so important in these cancers? So we looked at how important or what MLL does in cancer cells. So initially we did some cultured cells and knocked down MLL and started to look whether tumor cells grow or they die. And based on these, we find that if we knock down, we have done many experiments. Here is one where if you knock down only single gene, that histone methyltransferase, MLL1, in cultured cell, they die. This is a colon cancer, colon normal, breast cancer, breast normal, lung cancer, lung normal. You see the cells tend to die. So that kind of triggered that if the tumor cells are die when we knock down this MLL, can we try to see whether it kills the tumor. So this is a common preclinical model for studying this type of anti-tumor drugs, where you take a nude mice, implant the tumor under the skin. There are other type of xenograft as well. These are called xenografts. You see, within a month, the tumor started to grow very nasty. So this is the control, and when we knock down the enzyme, the histone methyltransferase MLL1, we did a systemic administration of an antisense oligonucleotide. So basically what it does, it knocks down the enzyme level in the body. So once that enzyme is decreased, you see we are able to arrest the tumor growth significantly. This is the uncontrolled tumor growth in the control animal, and this is upon treatment with the MLL1 antisense. I think this was a very dramatic observation that you are knocking down one gene and you have such a dramatic arrest on tumor growth in animal model. So we took out these tumors from the control and treated. We did all the pathological analysis. I don't want to go into detail today, but what they saw is that MLL1 is actually critical for the tumor growth, and it is involved in not only regulation of the tumor growth, it is also involved controlling the de novo angiogenesis. Angiogenesis means blood vessel formation. In a tumor section, pathologists look for blood vessels. If there is, for some reason, certain tissue has increased number of network of blood vessels, that means there is something wrong and potentially a cancer. And that happens in a lot of the tumor. So it happens that this particular enzyme, which is called MLL1, is actually required for formation of these de novo or new blood vessel inside the tumors, so that it can supply the oxygen for the growth of the tumor cells. So MLL is actually controlling the angiogenesis, and that's how it is controlling the tumor growth. When we inhibit the enzyme or knock down the enzyme, it interferes with the blood vessel formation, so tumor cannot grow. That's why we observe a dramatic arrest in growth. So now it's very interesting because we understand the pathology or biochemical signaling mechanism, but being this is an enzyme, a histone methyltransferase, now it a great news for chemists that now we can design molecules which can inhibit this particular enzyme, and that molecule could be potential therapeutics for variety of cancer. So this is actually the currently ongoing project that we are looking. Along with the biology, we are also looking at the MLL1 inhibitors for targeting some of these cancers. So this is one of the projects where we have spent some time. I don't want to spend a lot of time, but we also looked at, there are a couple of different MLLs. MLL1 is primarily involved in leukemia, but there are a couple of different MLLs, MLL2, MLL3, MLL4, MLL5. I think 2 to 5, they are not involved in leukemia. So they are likely important in other signaling process, and my lab shows that these MLL actually interact with nuclear hormone receptors, steroid hormone receptors like estrogen receptors, control the estrogen dependent gene activation and signaling. So steroids are critical for reproduction, development, all kinds of stuff, immune system and so on. So steroids actually control specific gene expression, and along with the steroid hormone receptor, in this case estrogen receptor, you need variety of other factors. So MLL happens to be the other factors, actually histone methyltransferase, which introduce methylation on the chromatin in response to hormone and turn on the gene expression. And we show that MLL controls variety of genes, and here is a gene called SRB1, which is involved in maintaining the plasma cholesterol level. In fact, studies from my lab shows that MLL2, one of the histone methyltransferase, actually critical for controlling the plasma cholesterol level. Part of these studies published in this particular article a couple of years ago. So at this point, my lab is going more towards translational. We have done plenty of cell culture, animal model, but until we can extend the study to the human, actually we have not done any, maybe some, but until and unless we can extend the study, make it translational, I think the job is not completed. So we started to look at the cardiovascular or metabolic functions in different humans. And this is a slide. I think all of us should appreciate what exactly it says. I'll go slow so that everybody can understand. What it says, this is from American Heart Association, this data. So this is true. So what it shows is the cardiovascular risk of cardiovascular disease as a function of age in male and female. As you can see, when the age is below 40, that means the woman, female are at I think the premenopausal stage or reproductive active stage. So at that stage, the cardiovascular risk for the female is much lower compared to the similar age men. And this risk goes high for the both male and female, and the difference between male and female actually goes down as the age, and it actually becomes almost same or even slightly higher at the postmenopausal stage. This is actually very, very important data because it says that during this premenopausal stage, when there is a hormonally active, that ovarian hormone like estradiol is giving a huge protection to the cardiovascular function. It actually controls the cardiac plasticity and vasodilation, all kinds of stuff. These ovarian hormone controls, but in the postmenopausal stage, this hormone level goes down, so woman starts to lose the hormonal control of this cardiac protection, and cardiovascular risk goes high. This is kind of well known, but how exactly this ovarian hormone controls the cardioprotection, it's still very, very unclear. So in my lab at this stage, actually we are studying how ovarian hormones control the hepatic metabolism. Liver is a very critical organ where all this lipid metabolism takes place, and so it appears that this ovarian hormone has a lot to do with the liver, the hepatic function, liver function, lipid metabolism, glucose metabolism, and so on. So at this point, a good amount of study in my lab is going on how ovarian hormone is controlling these functions of the hepatic functions. And towards that, we are actually drawing the blood sample from the mice, as well as we are also getting into human at different ages, and in mice we are able to collect the liver, but in the human we are only collecting the blood samples, and we are trying how exactly the different enzymes and liver is controlled, or different metabolic decisions are controlled by ovarian hormones. So so basically we are looking at how ovarian hormones controls this disease and looking at different human populations. So this is kind of the trying to understand the epigenetic signaling mechanism, how it controls cancer or metabolism, or how it controls the cardiac metabolism and functions. I want to switch the gear a little bit. Again, I want to be slow because I want to introduce a new field called non-coding RNA, which some of you or many of you know, but those of you who are chemists, biochemists who do not know, I think this is a very, very important area of research, along with all the biomedical research what has been done. So it is, I want to introduce this. So we are getting into something, function of this chromatin modifying enzyme and non-coding RNA in inflammation, immune response. It happens coincide with COVID-19 infection. So let's introduce what are non-coding RNA. Few slides, few minutes back, I described that we have DNA, DNA becomes RNA, RNA becomes protein. This is a kind of standard flow of genetic information. Protein gives phenotype. Human has about large genome, about 3 billion base pairs. And in 2001, when the complete human genome or nearly complete human genome was sequenced, and people realized that there is only about 20,000 protein coding genes present in human genome. So that 20,000 protein coding genes occupy only about 1.5% of the whole genome. So human genome is pretty big, and out of that, only 1.5% of the whole genome codes for the proteins. What is the rest of the genome doing? People did not have any idea. And people, because we did not have any idea, until about 10, 15 years ago, we all called them junk DNA. But all the scientists, although we called them a junk DNA, you have to understand that nature spends a lot of energy and time to replicate this big genome. If something is junk, have no function, it will not be carried out. It will be eliminated from evolution, genomic evolution. But as it is carried, even if we called them a junk DNA, it was not really junk. 2007, when the genomic information was, sequence information was uncovered, people started to dig into this junk DNA, what exactly this junk DNA contains. And that project called ENCODE project, Encyclopedia of DNA Element, and that project started to dig out the content of this junk DNA. And this study resulted that actually 80% of the genome, which we called non-functional or junk, they are actually functional. So 80-90% of the genome actually functional, and they actually codes for RNA. So these RNA, so genome makes the RNA, and these 80% of the genome which makes these RNA, and these RNA are not translated into protein. Even if they're transcribed into RNA, these RNAs are not translated into protein. That's why people have not discovered them. So 80 to 90% of the genome codes for RNA which mostly do not get translated into protein. Those are actually termed as non-coding RNA. So junk DNA is not really junk, rather it is a gold mine because it codes for these non-coding transcript. A couple of years ago, we wrote a review article, invited review article to summarizing these non-coding RNA. So we know that protein coding genes codes the protein. So their protein has certain function, something is enzyme, somebody is structural, and so on. But when the RNA is not translated into protein, what these RNAs are doing in the cell? And there are, unlike only 20,000, there are hundreds of thousands of these non-coding RNA in cell, and many of them you can detect now in the blood, you can detect in some of them we detect in the urine samples, what these RNAs are doing. So this particular review article actually summarizes that even if these RNAs are not converted into protein, they actually control the genome function. I want to explain this particular slide a little bit. If we consider, we actually represent the whole human genome as a musical instrument. Could be a harmonium, could be a piano. Okay, and if you play the piano, you will get a musical note. Sa, re, ga, ma, pa, all these, correct? Some music, some sound will come. And if you consider the human genome as a musical instrument. Notes are kind of protein products. And the non-coding RNAs, although they cannot be converted into the musical notes or protein products, they actually act as a player of the musical instrument. And the way they play, that type of signal or proteins are produced. So we represented non-coding RNA as kind of musical instrument player. It controls the, although it is not translated into protein, it controls the genome function, genome dynamics, chromatin dynamics, and that's how it controls the protein production, functions, and phenotype. So these non-coding RNA, which are not translated into protein, they actually have a critical role, and most of these functions are not discovered, yet to be discovered. So a couple of years ago, we started working on a non-coding RNAs. These non-coding RNA, I should say, they're expressed in all over the body, all the cells. Some of them have tissue specificity. They can be, some of them could be detected in the circulating fluid like blood. You can detect also some of them in urine. So because they're abundant, they have tissue specific function, they are potential biomarker. So we started working on a non-coding RNA, which was one of the earliest non-coding RNA, well characterized, called hot air. It was discovered based on its association with cancer. So it was overexpressed in cancer several thousand-fold, and it's a potential biomarker. And we started working on this because this actually regulates, it suppresses the expression of tumor suppressor. So when this RNA, it's about 2000 base pair long RNA, when it is expressed, it suppresses the tumor suppressor. If tumor suppressor is repressed, it contributes to tumor growth. And it actually, I don't want to go in detail, it interacts with some gene silencing, chromatin modifying enzyme called EGH2 and LSD1. So this RNA interacts with these protein silencing proteins and brings it to the chromatin and silences the gene expression. So in my lab, we looked at function of these non-coding RNA hot air, and we show that it is critical player in breast cancer. It is controlled by the steroid hormone estradiol, and it is actually epigenetically misregulated by endocrine disrupting chemical like BPA, which is a common contaminant in plastics, and there are some hormone therapeutic drugs which are really, really nasty. I don't have time to go. So we showed that this oncogenic RNA expression is affected by these endocrine disrupting chemical like bisphenol A. There is something called diethylstilbestrol, which is a drug used for induced fertilization and miscarriage, and this exposure to these estrogenic chemicals induce this expression of oncogenic RNA, and that contributes to cancer. So we published this article a couple of years ago that hot air is oncogenic RNA and its expression is controlled, interfered by BPA and DAS, which increases the risk of cancer. And this particular article was very much highlighted in variety of news, health news, and media. So we worked on this particular non-coding RNA for a couple of years, and in this particular story, we actually started looking at the function of this non-coding RNA on immune response. Now we are getting close to the COVID-19. I don't know how much time I have. Maybe another 15 minutes I will take to wrap up. So hot air, we started looking at immune response. We started looking at immune signaling. It's a very interesting signaling. So some of you are maybe experts, some of you may not know that when a pathogen like COVID-19 virus or bacteria infects, our immune system starts to work on it, and immune system has variety of cells, and they start to work on it, and like pathogen activates something called immune cells called macrophage. Macrophage activation takes place upon infection, and once the macrophage activation takes place, it starts to secrete called cytokines, chemokines, inflammatory gene which induces inflammation, and that actually cytokine levels, etc., inflammatory signal induces the increase in body temperature, fever, headaches, and all kinds of stuff, because your body or immune system starts to fight these and eventually kill these pathogen. So take home message is that when a pathogenic infection takes place, immune system, macrophage and other immune cells becomes active, and that releases these cytokines. And some of the cytokines are called interleukins, tumor necrosis factor, alpha, INOS. These are all pro-inflammatory genes, and sometimes, very interesting, these without infection or upon infection, these interleukins levels sometimes go few thousand-fold high in the blood. So by looking at the interleukin level, one can predict whether there is some infection and in the body. So this pro-inflammatory gene, the storm, is critical. Production is a signal for immune response, inflammation, and eventually pathogens are killed and taken care. It has other functions as well. So because there is a dramatic change in gene expression, and we work on the hot air non-coding RNA, which is a classical repressor, we thought maybe in the absence of pathogen, hot air is keeping this inflammatory cytokine level repressed. Upon infection, hot air is removed and repression marks are removed so that cytokine expression is induced. So with that hope, we started looking at the macrophage activation with the bacterial cells infection mimic with lipopolysaccharide. It is called LPS. It's actually bacterial cell surface extracts. So if you treat the macrophage, there is an inflammation induction. Cytokines are expected to go high. And we isolated the RNA. These are RNA sequencing. You see that interleukin level or INOS, these cytokines or pro-inflammatory genes are few thousand-fold high. And what we observed along with these known cytokines, hot air level is also significantly induced. This was kind of opposite to what we expected. And because hot air is working like cytokines, we knocked down hot air RNA in the macrophage and then looked at the interleukin level. And what we see that if you remove the hot air or knock down the hot air RNA level, the LPS induced inflammation or immune response is decreased. We looked at the signaling biology a little bit, and I don't want to go into detail. There's a transcription factor called NF-kappa B. And this NF-kappa B normally remains silent. Upon infection, the receptor signaling is triggered, cascade of signaling, NF-kappa B gets activated, and that NF-kappa B transcription factor binds to the promoters of interleukins and cytokines and turns on the gene. So what we observe is that hot air actually is playing a critical role, we don't know yet exactly how, in controlling these transcription factor NF-kappa B activation. NF-kappa B activation is there in any kind of infection, into COVID-19 infection. I'll talk about that host cell response. This transcription factor is very critical, and it looks like that non-coding RNA is critical player. So until recently, until last few years, people always thought protein coding genes like cytokines, these are the major players in controlling the immune response. But now we and other laboratories together demonstrate no, beyond the protein coding genes, which are only 20,000 in the cell, there are many non-coding RNA. And these non-coding RNA can also play a critical role in immune response, inflammation, and infection. So we looked at this biology and we show that this RNA is critical for cytokine expression by activation of NF-kappa B. Now that we show that hot air is required for inflammation, this did not satisfy everything. We work on hot air because we have been working, it is involved in cancer. Cancer is also inflammation. type of inflammation. But there are hundreds of other 100,000 other non-coding RNA. They may also be very important in inflammation and immune response. So hot air may not be the most important player. So at this point, we are trying to identify other RNAs along with the protein coding genes, what non-coding RNA may be involved in inflammation and immune response. What we did 2 rounds of experiment. We actually isolated the primary cells, primary bone marrow. From the bone marrow of mouse, we derived the macrophage, which is the immune cells, and exposed, stimulated with the inflammatory response, inflammation, and this time we isolated the total RNA, not the hot air. Total RNA means it contains the coding genes as well as non-coding genes. So total RNA from the cells, and there is a technique called RNA sequencing where you can actually identify the complete transcriptome. What are the gene expressed in this particular situation? You can identify both coding and non-coding. We have also done some work on cultured human cells, and we are also looking at the primary blood cells which are isolated from human patient and exposing to this inflammatory signal and looking at the transcriptome changes. And based on some of these studies, we actually identified whole transcriptome analysis. We identified a series of coding as well as non-coding transcripts which are linked to inflammation and immune response. They contains, I did not go into detail, but non-coding RNA could be long, pretty big, few thousand base pair. They could be pretty small. There is something called microRNA, which are about 22 nucleotide. So all different variety of non-coding RNA is there in our list. So we have actually a series of non-coding RNA which are up and down regulated upon inflammation. This is some of the selected top 10. You see the top 10 upregulated contents, interleukins which are expected in these systems, and there are many downregulated, many upregulated. There are microRNA, and there are many non-coding RNA. Problem with these studies, although they are upregulated or downregulated, they are closely related. One of the major problems is that non-coding RNAs, there's very little information on their sequence, structure, and function. So we don't know how big is the RNA, but all we can say that they are associated with inflammation. So it's good as well as bad. Good part that we have discovered something new. Bad part is that we have to start from the scratch to characterize, identify the function of this non-coding RNA. But here is we further analyze that this non-coding RNA, what we see on RNA sequencing, can also be reproducible in real-time PCR, qPCR, where this RNA is 350-fold, non-coding RNA is 350-fold overexpressed upon inflammation. Actually, this RNA, which are involved in inflammation, my laboratory has termed there is something called long non-coding RNA, inflammation associated RNA or LINF RNA. So LINF RNA, now onwards I'll talk. LINF RNA is a long non-coding inflammation associated RNA. Not long non-coding means their size is above 200 nucleotides. Now, at this point, what we have in the lab is that we have a few hundred to thousand of these LINF RNA. We term them human LINF RNA 1, 2, 3, and so on, up to 1,000. Here it is. Now, because these RNAs are associated with inflammation and immune response, we are trying to take the advantage to translate them as a biomarker. Even if we don't know their function, we can detect these RNA fragments in the blood cell, some of the RNA in the blood cell and some of them in the urine cells. So basically, one of my graduate student who is actually from Midnapur College, Abhishankar Chini, he started this project significantly in deep. So what he is doing, he has actually created a non-coding RNA array. So basically 96-well plate. each well will be representing a particular non-coding RNA. So this non-coding RNA array will be probed with the blood samples, RNA isolated from the blood samples from different patients. It could be patient who has autoimmune disease, it could be patient who has bacterial infection, meningitis, or it could be patient who has COVID-19 infection. So if we isolate the blood sample, RNA from the blood sample, these 96 or 386 non-coding RNA array can be probed to see what non-coding RNA is overexpressed there. So based on that, we can actually select few non-coding RNA, which we call lymph RNA, for developing potential biomarker for a particular type of disease. I skip that. So now I have another 2-3 slides to talk. Now we have a project going on currently to look at, because of the COVID-19 pandemic, we started to look at the inflammatory, because we are already working on inflammatory signaling. We have a series of novel RNA, non-coding RNA, which are closely linked to inflammation. Now we can try to see what is some of these non-coding RNA is also involved in COVID-19 infection. Can we utilize them to diagnose the COVID-19 infection or severity of the disease, or predict the infection severity and risk of the disease in human population? So this is a kind of slide. I did not have much time, so I took this slide from YouTube. There are a lot of nice videos how exactly COVID-19 SARS-CoV-2 works, how it infects the human host cells, how it virology, and how it is packaged into variety of new virus particle and released into the atmosphere. So there are actually a lot of excellent articles to give the virology. So virology, surprisingly, only less than a year, a ton of virology about the COVID-19 has come out. Basically, virus enters into the cell with a receptor called ACE2, angiotensin converting enzyme. Two is angiotensin receptor, which are linked to hypertension. That's why the hypertensive population are at more risk of COVID-19 expression. Also, I think male, this ACE2 is expressed in the males. So that's why I think males get more affected. It is coming from the X chromosome. Males get more affected to this COVID-19 infection compared to female. So virus enters into cells. It is a RNA virus. RNA is duplicated and eventually packaged into new virus. And very interesting for this virus, when virus is released, it has a kind of lipid coat around this. So that's why I think washing hands with soap actually significantly help removing of this virus or inactivating this virus. So there are a couple of things is very, very interesting, very unique for COVID-19, that compared to other viruses, this has very high titer. So that kind of virus load is very high. That increases its risk of transmission, infection, and so on. So I think because of this vesicle around the virus, it's good that we clean up the hands or sanitize our hands with alcohol to destroy the lipid and inactivate this virus. Now, people, when they get infected in 3 to 6 days, the people starts to get symptoms, the fever, headache, and all kinds of stuff. And eventually, for some people, depending on the person, depending on the treatment, and most people survive, and some people are vulnerable to this particular infection. One of the things which I want to talk is something called cytokine storm during the COVID-19 infection. It's a very, very important phenomena that when COVID-19 infects the host cell, humans, our immune system starts to react. Immune system starts to work, and it starts to release the immune responsive, activate the macrophage, T cells, and all kinds of immune cells, and starts to release inflammatory cytokines, pro-inflammatory cytokines like interleukins, IL-1 beta, INOS, interferons. All these cytokines and interferons are released into the bloodstream, and that to fight the COVID-19 infection. But one critical thing happens in cytokine, because whatever the reason, the cytokine production upon infection of this COVID-19, cytokine production keep on increasing and to a high concentration. And that's called cytokine storm, and many of these cytokines expression normally should go down when the virus titer should go down after a couple of weeks, but it appears, it's not, total picture is not clear. It appears that many of these cytokine level do not go down even if the virus titer is goes down. So there's a boost, there is a storm, huge high increase in cytokine concentration. So high concentration of cytokines are produced. But this is a major factor for the outcome of the patients. The people, this high cytokine kind of induces the huge inflammation in the body. So it is linked to heart malfunction, this high cytokine level, heart malfunction, liver malfunction, gastric malfunction, brain malfunction, and so on. So take home message is that COVID-19 infection is a problem because people die acute respiratory syndrome when the virus infection is going on, but when the virus infection is low, has gone down, still the cytokine level has not gone down. Huge cytokine level is still there in the bloodstream, and these high cytokine level for long time actually contributes the multiple organ malfunction. So even if, so actually lot of time a person did not die out of COVID-19 infection, but they die out of heart attack. It is because that the cytokine storm resulted malfunction of this cardiac muscle and all this stuff. So the post-cytokine COVID-19 infection cytokine storm is playing a critical role on the severity of the disease. So a lot of studies are right now trying to understand the different cytokine concentration across the lifespan of the virus, even after the virus titer is gone down a couple of weeks. Still people are sick, even the virus titer is down because of the cytokine storm, and identification of these cytokines correlating to the disease stage, severity or morbidity or organ malfunction is very, very critical, becoming very critical in determining factor what kind of therapy we can design. So a lot of studies are focusing on identifying the cytokine storm at different stages of COVID-19 infection. Now this becomes a very interesting subject for us because cytokines are protein coding genes, but we know that protein coding genes alone are not enough, not all. There are tons of non-coding RNA which we termed actually long non-coding inflammation associated RNA or lymph RNA. In my lab, we have at least a couple of hundreds of these lymph RNA we have identified which are linked to inflammation. So at this point, what we are doing is, I could not disclose some of these studies. What we are doing that we are using our lymph RNA array to probe with the blood sample from these COVID-19 infected patients at different stages and see, along with the pro-inflammatory cytokine, interleukin, INOS, IL-1 beta, we are also trying to identify what lymph RNA or what non-coding RNA are critical to COVID-19 infection, how they can be correlated to the stage biomarker, novel non-coding RNA-based biomarker, how they can be correlated to define the stage of the disease, the potential outcome of the disease recovery. And also there is lot of studies, the different ethnic population, male, female, different genetic background, people are at different risk. Can we correlate based on these coding as well as these lymph based biomarker? Can we correlate or define the stage of the disease or biomarker or outcome of the disease that can guide the potential therapeutics? So this is where I think my lab is looking for next couple of weeks and months. So these are all biology, and as I said, I am a chemist. My eyes are always on molecules. In parallel, we are also screening for different types of molecules for epigenetic inhibitors for the potential COVID-19 infection. We are looking at anti-inflammatory molecules in our inflammation assay, or some of them steroid hormone analogs, which are critical in modulating the immune response. I think that's all we had. So couple of projects we discussed. Epigenetic, I hope I have introduced the concept, basic concept of epigenetics and how the MLL controls the cancer and things like that. How the non-coding, I also defined what is non-coding RNA, how these non-coding RNA could be exploited to develop potential biomarker for a variety of disease. We are focused on inflammation, immune response at this point for COVID-19 detection, and looking at, I termed it as a lymph RNA storm. That was the term from my lab. So this is my group. I think many of them has graduated with PhD and they are gone, and there are some students. I mean, one of my current student is actually from Midnapur College, joined 2 years ago. Last year, I recruited many students from India, and so in our department, if any of you are interested in future doing the research in chemistry, biochemistry, you are welcome to contact me. I can help and guide. At least we can advise what steps you may need to do. My lab is funded by National Institute of Health. Some of the studies, cardiovascular stuffs, are funded by American Heart Association, partly by National Science Foundation, and by the University of Texas at Arlington. With this, I would like to thank all of you for listening to me. I am ready to take questions now, if you allow, or you can always approach me later by email or phone or whatever it needs to be. Thank you again, and thank you, Dr. Guchait, for inviting me for the presentation.
Thank you.
Thank you, Professor Subhranshu Shekhar Mondal, for your precious time and your presentation and enlightening us regarding present advanced research in your laboratory. You have nicely shared your work with us. It would be helpful to us and our students to understand ACC and DACC papers of the new CVCS curriculum of our university, which has included biology and others interdisciplinary subjects. You have also well explained the terms like genetics and epigenetics, gene regulation, mixed lineage leukemia, gene regulation in different diseases, angiogenesis, risks of cardiovascular disease, role of non-coding RNA in inflammation. And also you have well explained the therapeutic design. Now, here is a question regarding your presentation, where you have said that no non-coding RNA hot air is a tumor suppressor. what happens if it is presenting in term or in Jang Jing? Will it still be effective?
Okay, I think I did not go over detail. What are the non-coding RNA? They can be originated actually from different region. Like many non-coding RNA are present in introns. Many is present in exons. Many of them are intergenic. Okay, in between the gene. Also, I think very interesting to know that the protein coding genes are in one strand, which is called the sense strand. We have two strands of the DNA template. Protein coding genes are sense, and many of these non-coding RNA, it happens that they're antisense. So the other strand of the genome, which is antisense strand, is also equally important for the genome function. Now there are different types of non-coding RNA. They could be intronic, because a lot of time people thought that introns are useless only for diversity, but we know now that they're actually critical source of RNA which may regulate that particular gene. So many of them could be replacing tumor suppressor, like they're like protein coding genes. They can interact with other proteins because they are nucleic acids. So they can replace the tumor, they can activate the gene, they can contribute to tumor growth, they can contribute to immune response. Depending on what exactly we are doing. Whatever happens to be a repressor, but in our study we find it is also an activator, especially in case of immune response. It is required for the activation of the immune response. I don't know whether I answered the question. Yeah.
No, right.
Thank you.
It's clear to me. Another question: the mutation of gene in childhood is dependent on male or female? Mutation of gene in childhood. Yes.
It can happen both. It can happen to both male and female. But what drives this mutation is especially I think you may be talking about the MLL, the mixed lineage leukemia. It can happen both male and female. But what drives this mutation is not clear, but maybe some chemical stress, environmental stress may be a factor, depending on how they're exposed, air quality, water quality, or how much drugs, hormone therapy. So some of these together, chemical stress or radiation, or maybe genetic contribution to the genomic instability, all of these factors may contribute the mutation. But they can happen both male and female.
Thank you for enlightening us. Very much. We are grateful to you.
Thank you so much, Dr. Buchait. And I think it's a great initiative what you all are doing. And I'm glad that I could share some of my thoughts with you all, and especially the students. And that's why to give thank you. So I leave the stop sharing the screen and end the call, or
okay, you can leave them.
Okay, thank you. And when you come back, because I want to hear the Dr. Jana, Malavindu Jana, and others. So can I join them?
Yes, yes, of course. I will send a link.
Okay.
5:30, I will send you the new link.
Okay, thank you so much.
I will send you the new link.
Okay, thank you so much. I appreciate. Okay,
thank you.
Thank you.
Now, there is another question to Dr. Dipankar Mondal. To Dr. Dipankar Mondal, can you hear?
Yes, sir.
Dr.
I can.
Dipankar Mondal.
I can.
Okay. Yes. Here is a question. You explained us about how alcohol consumption affect at molecular level. You also said that how research is going in going beyond the heredity of 1 minute. Yes, but what about the genes of alcohol consumption which are present in the junction and removed during splicing? Will they also be inherited? Can you hear?
Yeah, I can hear. My answer would be like, I want to answer. I don't know exactly. I can satisfy the who has questioned this. The alcohol only increase or decrease gene expression through histone acetylation. As you see my presentation, alcohol finally from aldehyde to acetate to acetyl CoA, then finally histone acetylation. And this histone acetylation is like one of the major factor for increasing gene expression because it loosens the DNA wrapping around the histone protein. So DNA are now free, RNA polymerase can be recruited, and the gene expression can happen. And there are a lot of studies and very well-reputed journal. You will find the paper in Nature journal also. The histone acetylation has direct connection with gene expression. And this paper also has presented some of the data that there is increase in gene expression. Some of the gene has also decrease in expression. So out of 2000, they have measured RNA-seq data they presented. I did not presented that data because it's a bit critical. It's a not to present for the general audience. It's a particular specific people who are expert. I am also not expert RNA sequence experiment. I only can judge the result. I never did the experiment, that I only interpret the result. So that RNA-seq method they presented in that paper, you will find I presented the Nature paper in 2019. So there is so that out of 3,613 genes, there are 2,107 genes which are affected by this acetate. So that does mean there is no control. It can increase or decrease any of the gene. So that's not a good factor actually, because histone acetylation as increases gene expression of your good gene, some also bad gene. And the junk gene we are talking about, the splicing, I don't think it has any direct relation with acetylation. Splicing is a different process during mRNA to for protein coding, that intron or exon, they are the non-coding are removed and the mRNA are produced. So I don't know there is any direct relation with histone acetylation with that splicing. So I don't think that acetylation will directly can affect on the junk gene or not. That would be my answer.
Okay, thank you for your explanation. It is clear to me, partly clear regarding the question. Thank you.
Okay.
There is no question to you. Thank you once again for your presentation and for your presence, kind presence.
Thank you.
Now I move on to our colleague Samirul Islam.
We are thankful to all our respected speakers, all teachers and students who are participating in this webinar. Now we are ending the first session of the webinar. Our second session will start at 6 p.m. We are end our broadcast now. Thank you all.